The article that we decided to do our research on is called Breakthrough in Salt Tolerance in Plant Research, which was published September 20th, 2016 at the University of Adelaide.
Hello and welcome to the show. Tonight our special guests are representing their research project in salt tolerance in plants. Today our special guest is Dr. Cloretta. Hello, Dr. Cloretta.
Hi, how are you?
Good, how are you?
Good.
So I hear you and your partner have been doing some research in correlation to plants.
Yeah, it's specifically about our breakthrough in salt tolerance. We recently discovered a new protein that's also found in animals. It's found in plant cells. It's called the aquaporin, and it's a double-barreled protein that can transfer salt ions as well as water. Previously, we thought that the proteins, we knew they existed in the roots of plant cells, but we thought they only transported water. So this discovery is big because it helps us to explain how the salt gets into the plants. It also leads to new research that could be done in how the blocking of the pathway of the salt into the plants could be affected. It's going to be a good discovery.
And you and your partner have been working on this for a while now?
Yes, Dr. Florence and I have been working on it since we got out of undergrad. We both went to SUNY Oneonta. We actually met freshman year.
Oh, wow.
Yeah, Dr. Zalatan's class. We were friends and we went through all the science together. So we've been working together for a while, but we've been working on this project specifically for about 4 years.
Now, Dr. Cloretta is going to explain some of the background information towards the research that they have conducted.
Okay, so what we have up here is there's two forms of transport, active transport and passive transport. Active transport requires energy in the form of ATP, and it moves substances from a low concentration to a high concentration. But what we're going to focus on is passive transport because that's what we have going on in the plant roots. So passive transport doesn't require energy because we're moving from a high concentration to a low concentration. So what we have here is particles in a high concentration coming across the membrane into an area of low concentration, and this is known as diffusion. Another special form of diffusion is osmosis, and that's the transport of solely water, which is what we originally thought was happening with the aquaporins. Over here is facilitated diffusion. We have discovered, since the salt ions are moving through the membrane as well, these right here, this is the aquaporin, and it's still moving from high to low concentration, but with the help of a protein. No energy is required, but the salt ions will be moved through the membrane with the help of the aquaporin protein to get to the low concentration.
Welcome back. Now we are going to have Dr. Cloretta's partner, Dr. Clorence, join us to tell us a little bit more about his plant research. Welcome, Dr. Clorence.
Thank you.
How are you tonight?
I'm good. How are you?
Good. So what first got you interested in plants?
Freshman year at SUNY Oneonta, I was in Bio 180 in technically Dr. Zalatan's lecture, but Dr. Withington, who was the plant expert, got me interested in diffusion and active transport, and I wondered how stuff worked in plants, so I decided to go along with it.
Oh, cool. Now Dr. Clorence is going to explain a little bit more about the research.
So what we have here are plant cells in 3 different types of solutions: hypertonic, isotonic, and hypotonic. In the hypertonic solution, which is really unfavorable for plants, the water is leaving the cell because there's a higher concentration of salt or any other kind of solution outside the cell. Isotonic, there's no net flow of water because the concentration of solution is the same on the inside and the outside. And the favorable condition, hypotonic, there's more solution inside the cell than outside, so the water's going to rush in, keeping the plant turgid. Before we knew that the aquaporins could move salt, we wouldn't know how the salt would leave the plant cell in the hypotonic situation.
Thanks to Dr. Cloretta and Dr. Clorence's research, we now know that salt enters and exits the plant cell through channel proteins known as aquaporins. These aquaporins help keep the plant turgid in a hypotonic solution. Thanks for watching, and I'll see you next week.